Welcome back, children. Now I am going to deal with 5 different organelles, namely endoplasmic reticulum, Golgi Apparatus, Lysozymes, Ribosomes and vacuole. Same chapter, cell, the unit of life. So first let me deal with endoplasmic reticulum. As I am dealing with each organelle, I will be first emphasizing the structure and then the function. So first let us see the structure of endoplasmic reticulum. Endoplasmic reticulum is a network of tiny tubes or reticulum of tiny tubes scattered in the cytoplasm. It is running from the nuclear membrane to the plasma membrane. So it is stretching from the nuclear membrane to the plasma membrane. The tubes which are close to the nuclear membrane are associated with ribosomes and they are called RER, and the tubes that are towards the plasma membrane are devoid of ribosomes and they are called SER. RER stands for rough endoplasmic reticulum and SER stands for smooth endoplasmic reticulum. So we can divide endoplasmic reticulum into two types, RER and SER. So once again, endoplasmic reticulum is a network of tiny tubes or reticulum of tiny tubes scattered in the cytoplasm. It is stretched from the nuclear membrane to the plasma membrane. The tubes close to the nuclear membrane are associated with ribosomes and they are called rough endoplasmic reticulum, and those tubes which are away, that is towards the plasma membrane, are devoid, are not associated with the ribosome and they are called the smooth endoplasmic reticulum. Now this endoplasmic reticulum or the system of network of tubes divides the cytoplasm into two spaces, luminal space and extraluminal space. Space is also called compartments. So the space within the endoplasmic reticulum is called luminal space. The lumen of the endoplasmic reticulum is called the luminal compartment, and that in the cytoplasm, that is outside the endoplasmic reticulum, is known as extraluminal compartment. So we can see two compartments within the endoplasmic reticulum. That space is called luminal compartment, and outside the endoplasmic reticulum, that is the cytoplasm, it is called extraluminal compartment. Next aspect, function. Function of the endoplasmic reticulum. As I told you initially, there are two types of endoplasmic reticulum, RER and SER. The RER is associated with ribosomes and hence involved in protein synthesis. So the function is they are involved in protein synthesis, whereas SER is devoid of ribosomes and they are involved in lipid synthesis. That's all. So once again, endoplasmic reticulum, the structure, they are a network of tubules or network of tiny tubes or reticulum of tiny tubes scattered in the cytoplasm, stretching from the nuclear membrane to the plasma membrane. You can identify two types of endoplasmic reticulum. The tubes towards the nuclear membrane are associated with ribosomes and they are called the rough endoplasmic reticulum, and the tubes away, that is towards the plasma membrane, are devoid of these ribosomes and they are called smooth endoplasmic reticulum. This endoplasmic reticulum divides the cytoplasm into two compartments. The space within the endoplasmic reticulum is called the luminal compartment, and the space outside, that is the cytoplasm, it is known as extraluminal compartment. Talking about the function, RER is involved in protein synthesis and SER is involved in lipid synthesis. Next organelle is the Golgi Apparatus. The Golgi Apparatus consists of a stack of flattened disc-shaped sacs. Flattened disc-shaped sacs which are arranged parallelly to one another. See, one above the other, and you can see the sacs taper at the apex. Okay, and the location of this, it is when we look at the orientation of this, is concentric to the nucleus. If this is the nucleus, how is this going to be aligned? This will be aligned like this. See, concentric to the nucleus. See the alignment? So, once again it consists of a stack of 7 to 8 flattened disc shaped sacs. Each sac is known as cisternae. So, each sac is called cisternae. Now, its orientation is how it is located in the cytoplasm. Its orientation is, it's like concentric with the nucleus. See the way I have oriented it. It cannot be like this, it cannot be like this. It should be like concentric circles with respect to the nucleus. And another thing you should keep in mind, it is also found closely associated with the endoplasmic reticulum. Whether it is RER or SCR, it is very close to the, physically close to the RER and SCR. Clear? So, once again, Golgi apparatus consists of a stack of flattened disc shaped sacs. There will be 7 to 8 sacs arranged parallelly to one another, and this orientation is concentric to the nucleus, and it is closely associated with the RER and SCR. Now, there are two phases. You can distinguish two phases, the cis phase and the trans phase. The cis phase is a phase in which the materials enter, and trans phase is a phase at which the materials exit. So, to understand this entry and exit, we have to learn the function of Golgi apparatus. So, next we learn function. The function of Golgi apparatus is, just remember the short form PMD, that is, it is involved in packaging, modifying and dispatching of materials. For example, if it is the proteins that is synthesized by the RER, the proteins get in, I am giving yellow color for the proteins synthesized by the RER. These proteins get in at the cis phase. See, this is the entry. Actually, they enter through the cis phase. Now, in the cis phase, there are the cisternae. So, it enters the first cisternae. From the first cisternae, it gets into the second cisternae. Second cisternae, it gets into third. From third, it gets into the fourth. From the fourth, it gets into the fifth, and then it gets into the sixth cisternae. So, as is it moving from one cisternae to the other cisternae, it is getting packaged. How is the packing done? It is getting covered with a membrane, and it gets modified, and finally it is dispatched. How it is dispatched? It gets pinched off as vesicles. See, it gets pinched off as membrane bound vesicles. This is the way it is getting dispatched. So, the basic function is packaging. How it is packed? In the form of membrane bound vesicles. Then it is modified. I told you we are taking proteins. The raw material was protein, but when it comes out, it becomes glycoprotein. So, it gets modified and it is dispatched. Where it is dispatched to? It is dispatched either to intracellular targets or outside the cell. Clear? So, remember P, packaging, modifying and dispatching the materials, where within the cell or outside the cell. Now, usually it is packaging, modifying and dispatching what? The products of the RER and SCR. Products of the RER is the proteins, product of the SCR is lipids. So, if suppose I indicate lipids with another color, say pink color, see, lipids enter the cis phase. From the cis phase, it gets into the first cisternae to the second cisternae, third, fourth, fifth, sixth. As it enters from one cisternae to the other cisternae, what it gets in? Packaged, modified, and finally, when it reaches the last cisternae at the trans phase, it gets pinched off as a vesicle, membrane bound vesicle. And this vesicle, now it's not the initial lipids, becomes glycolipids. So, we have glycoproteins and glycolipids synthesized. The raw material is lipids and proteins synthesized by the SCR and RER respectively, but once they pass through the Golgi apparatus, that is from one cisternae to the other cisternae, and it is exiting from the Golgi apparatus in the form of vesicles, they are dispatched in the modified version, packaged, modified and dispatched in a new version, glycoproteins and glycolipids. Not only glycoproteins and glycolipids are synthesized by Golgi apparatus, even the lysosomes are also synthesized by this way, by the Golgi apparatus. So, I hope you have understood the structure and function of Golgi apparatus. What is Golgi apparatus? It consists of a stack of flattened disc-shaped sacs arranged parallelly to one another. There is a cis phase and trans phase. Cis phase is the phase through which the materials enters, and trans phase is the phase or the cistern from which the materials exit, and it exit after PMD, packaging, modifying and dispatching. Then the function of the Golgi apparatus: packaging, as I told you, PMD, packaging, modifying and dispatching to targets within the cell and outside the cell, and this is how glycoproteins and glycolipids and lysozymes are synthesized. Clear? And I told you very importantly, when you locate the Golgi apparatus, you cannot place it anywhere. You have to place it concentrically to the nucleus. So, the orientation should be in concentric to the nucleus, and it is closely associated, physically associated with the RER and SER. Next organelle is the lysozymes, and I told you that just previously it is synthesized from the Golgi apparatus. So, definitely it is a membrane bound vesicle synthesized by the Golgi apparatus. Only thing, the content, what it contains is the hydrolytic enzymes. Hydrolytic enzyme belongs to a group of enzymes called hydrolases. And there are different types of hydrolases depending upon the substrate: lipases which digest lipids, proteases which digest proteins, carbohydrases which digest carbohydrates, and also nucleic acids. So, once again the structure of lysozymes: they are membrane bound vesicles synthesized by Golgi apparatus. It consists, but what it consists? It consists of hydrolytic enzymes, hydrolysis like lipases, proteases and carbohydrases. Now, the function, very popular function. It is, with respect to its function, it is given two nicknames: the waste bin or the waste disposal system, and second, the suicidal bag. Why it is called the waste disposal system? Any organelle, be it Golgi apparatus or endoplasmic reticulum or vacuole or plastid or any organelle within the cell, if it becomes worn out or if it gets damaged, what happens is this lysozyme, it is a vesicle filled with hydrolytic enzyme, will rupture and release this hydrolytic enzyme, and this enzyme will digest that organelle. So, it works like a waste disposal system. Now, sometimes the cell may be damaged. In that case, what happens is rupture and releases the enzymes, and the entire cell in which the lysozyme is present will be destroyed. In that context, it is called the suicidal bag, because even the lysozyme itself is getting destroyed. I hope you understood the function of lysozyme. The next structure is vacuole, and you can identify the vacuole. Where is it located? In plant cell, it occupies nearly 90% of the volume in plant cell. Whereas in animal cell, they are very tiny and no significant role. Now, this vacuole in plant cell, it stores a lot of ions, Also, it stores water, excretory product and cell sap in plant cells. So, in plant cells, they are having a role to play, as well as in protistants, like amoeba also, they are very important, they play an important role. Whereas in animal cell also it is present, but no significant role, they are very tiny and there is no much significant role. Okay. So, once again describing the structure of a vacuole. Vacuole can be described as membrane bound space. It's just a space. So, what is a vacuole? It's a membrane bound space. So, definitely there should be a membrane. See this blue color? That's a membrane, and this membrane is called tonoplast. This tonoplast is very unique. It has a unique property of allowing ions to get collected in the vacuole. Usually, please focus here, usually if these are two regions, see lot of positive ions and this region with few ions. So usually there is a tendency of movement of ions from a region of high concentration to a region of low concentration. That's a usual tendency. In water also, movement of water from a region of high concentration to a region of low concentration is called osmosis. So that is the usual tendency. That is called passive transport. Clear? It's through a semi-permeable membrane. But here in tonoplast, the tonoplast is unique. It allows the reverse movement of ions from a region of low concentration to a region of high concentration against the concentration gradient. So that is the property of this tonoplast, and hence the vacuole in plants is richly accumulated with ions, a lot of ions, positive ions and negative ions. They are richly supplied with ions because the property of the tonoplast to enable diffusion of ions, or to enable ions against the concentration gradient, that is from a low concentration to a region of high concentration. So once again, the structure of vacuole, I am emphasizing this, membrane bound space which occupies in plants 90% of the volume. In amoeba they have some role to play, as well as in animals they have no significant role, but they are present, they are very tiny. Okay. Now talking about the function. When we talk about the function, I am dealing with plants and protistans. Now in plants, we have learnt in 10th standard, it stores water, cell sap and excretory products. Very, very important. Excretion is also, the excretory product are stored in the vacuole. Okay, and also very importantly, as we said, it stores a lot of ions. So once again, in plant cells it stores water, cell sap, ions and excretory products. And because of this nature, that is, it's fully turbid, filled, it gives rigidity and strength to the plant cell. Okay, we know rigidity is provided to some extent by the cell wall. Also, this plant cell is always turbid or strong because of the vacuole, which is fully filled with ions, water, cell sap, etc. Okay, it's never flaccid. So it provides to some extent rigidity to the plant cell. And very importantly, it stores excretory products, cell sap, ions, water. Now in animals like amoeba, we know now amoeba is coming under protista. There are two types of vacuoles, namely the contractile vacuole and the food vacuole. Contractile vacuole and the food vacuole. Contractile vacuole is involved in excretion of waste, and food vacuole is involved in digestion of food. So there are two types of vacuoles. One is involved in the excretion of waste, and that is called the contractile vacuole, and the food vacuole is involved in the engulfing of food and digestion, and that is known as the food vacuole. In animals, though they are present, they have no significant role. Clear? Vacuoles. And last organelle for this video is ribosomes. And in the beginning I told you the specialty of ribosomes, they are non-membrane bound organelles, and to make it complete, in the case of organelles I am going to write non-membrane bound granules. They look like granules. Only here you can see two structures, but actually under the electron microscope they look like granulated structures having two subunits, the large subunit and the small subunit. So once again, talking about the structure of ribosomes. Ribosomes are non-membrane granules having a large subunit and a small subunit. And we know there are two types of ribosome, 80S ribosome and 70S ribosome. 80S found in eukaryotes and 70S found in prokaryotes. It's indirectly a measure of density and size. So from this what we can infer is eukaryotic ribosomes are denser and larger than the prokaryotic ribosomes. These ribosomes are made up of proteins and ribonucleic acid. So once again, talking about the structure of ribosomes. Ribosomes are non-membrane bound granules. We have two subunits, namely large subunit and the small subunit. Though they have, you can't see that bifurcated version, you see it granular under the electron microscope. There are two types of ribosomes, 80S and 70S. 80S is found in eukaryotes and 70S is found in prokaryotes. Now function of ribosomes. The function of ribosome, very carefully you should remember, the function is just they serve as sites for protein synthesis. they just serve as a site or platform for protein synthesis. They are not synthesizing, but they serve as sites for protein synthesis. I hope you have understood these 5 organelles that I took in this video. Learn it under the heading structure and function. Thank you.